Okay, hey team, welcome back. Cloning Part 2, Modern Methods. In this screencast, I'm going to talk to you about how we can create a clone from differentiated cells. As you recall, last week we talked about, or Chang talked to you about pluripotent cells. So an embryo really early on in development can be split, or a cell can be plucked off of that embryo, and you can grow or you can clone a new organism from that early mass of cells. And the reason why that's so easy to do is because the cells are all pluripotent. They haven't picked a certain destiny or a certain fate yet. It's like a kid really early on in their life. They haven't picked a career path, and it's really easy to change and become anything. Now, cloning an organism, an entire organism, from cells that have already been, or a cell that has already been differentiated, is a bit trickier, but also a bit more useful to us. And let's talk about the reasons why it is a bit more useful. So imagine we have a cell here, and this is a skin cell from a cow. Oh God, this is a terrible cow. Okay, let's give him a spot, and then another spot, and some ears. Okay, this looks a little bit more like a llama than a cow. Okay, terrible cow, but you get the point. And this cow produces a ton of milk, and so it would save us a lot of space in the barn if we could produce, if we could have only this cow. Say it produces twice as much milk as every other cow. We could double our output of milk if all of our cows produced milk like this cow. But the problem is that this cow is already an adult, and so how do we take, for example, a skin cell from this cow and make it into, or clone, an entirely new organism with the same milk producing potential? And so the guy who first figured this out, or how to do this, or he was the head of the lab that cloned the first mammal, was this guy named Ian Wilmot. Oops, not Ian. So Ian Wilmot was the principal investigator at a lab at the Rosen Institute in Scotland. And in Scotland, they've got a lot of sheep, and his lab was trying to clone a sheep, and they succeeded in 1996. So let's break this down. So they cloned a sheep, and you might have heard of the sheep before. She's a pretty famous sheep. Her name is Dolly. Well, Dolly, let's call her Dolly 2. This is the clone of the sheep, or the clone that we all know and love so well. Well, she came from another sheep, or from the cell of another sheep. Let's call that other sheep Dolly 1. Well, here's the story about Dolly 1. Dolly 1 was actually euthanized, and that means killed, put in a minus 80 degree freezer, or all her different parts were. And so Dolly 2 was actually cloned from the mammary cells. Oops. Dolly 2 was cloned from the mammary cells of Dolly 1. And if you remember here, mammary cells are basically the breast tissue of Dolly 1. And these are somatic cells. Somatic cells, they're not egg and sperm, and they are fully differentiated. So how did Ian Wilmot and his team do this? Well, first they took a cell from Dolly 1, one of her memory cells, so let's draw it right here, and they enucleated it. So they grabbed the cell's nucleus and took it out. So they had all of Dolly 1's genetic material out here, and this, remember, this is 2N, so complete set of chromosomes. They had this genetic material outside of the original memory cell. Then they took another cell here, and this was an egg cell from another sheep. We can call her Lucy. I don't know what this sheep's name was. So egg cell from an entirely new sheep, and they enucleated that as well. So took the nucleus of this egg cell out. Remember, this is 1N here, so half the genetic material in the egg cell. But also in the egg cell wasn't just genetic material. Remember, there are all these factors, proteins and signaling factors that are going to be important in development. So those were left inside the egg. So what did they do next? Well, they took this 2N nucleus of Dolly 1's and they embedded it just below the membrane of this egg cell with all these factors. And then they zapped it with electricity. And it caused the cell to incorporate the nucleus into the cell. I don't know if I can still grab this. So, okay, that didn't work. Okay, so zap the cell with electricity, incorporates the nucleus, and now this cell is 2N. Now here's where Ian Wilmot's team did something completely different. No one had done this before, and this was the key element in cloning this adult differentiated cells. Because remember, this nucleus here is completely differentiated, and if left to just do its own thing, this will go on to become a bunch more memory cells, which doesn't do us much. It's not going to develop into a full organism. So what did they do that was so new and worked? Well, they starved this cell. They starved the egg cell. They took away some of the necessary nutrients in order for it to grow, and the cell undifferentiated. Essentially, when cells are starved, all the DNA turns back on. And so they were left with this egg cell that was now pluripotent again. So they took this egg cell. I'm going to select this whole thing, copy. Okay, so they took this undifferentiated cell, which now, remember, it has got the nucleus of Dolly 1, it's the egg cell from the sheep that we're calling Lucy, and they stick this cell into another sheep, the surrogate mom, you can call her Judy, I don't know what her name was either, and in Judy, this cell that has Dolly's genetic material starts to grow and develop into an embryo. Okay, I got tired of trying to put nuclei in each one, but you get the point. So, development, and when this embryo comes to term, out pops Dolly 2, the genetic clone of Dolly 1. Now, I'm going to show you a picture of Dolly. So here's Dolly the sheep as a little baby. So this is Dolly 2 right here, the clone, grown from the memory cells of Dolly 1 from the freezer. And here's the surrogate mom. I think we're calling her Judy. She's the one who gave birth to Dolly 2, but again, no genetic relation at all to Dolly. And so Dolly actually grew into a pretty famous celebrity, and you can see her here meeting the press. So this was the first mammal to be cloned in this way. And we'll talk a little bit more about this process and the implications of the power that came with the ability to clone mammals in class a little bit later on. Okay, that's it, and have a good night.